You know, when we think about the deadliest things on the planet, our minds, they usually jump to the spectacular, right?
Absolutely.
We think of, I don't know, a great white shark breaching the water, or the inland taipan snake out in the desert, maybe even the mosquito, carrying malaria,
Things with teeth.
or fangs.
Exactly. Things with teeth or venom sacs or wings. We think of predators. Things that actively hunt us. We very rarely think of something that just sits perfectly still in the dirt.
Doesn't make a sound.
No sound, no brain, and, well, frankly, it looks kind of beautiful.
It is elegant. That's the word I always come back to when I see one in the field. It's statuesque. It has this clean, almost architectural look to it.
And that elegance is the whole problem.
That elegance is exactly what makes it so incredibly dangerous. It doesn't look like a monster. It looks like a masterpiece.
We are talking, of course, about Amanita Phalloides, better known, I guess, by its heavy metal stage name, the death cap.
And I was looking at the numbers from our stack of research here. We've got medical toxicology reports from Stat Pearls. We've got genetic studies from the Pringle lab at the University of Wisconsin-Madison, even historical archives going back to the Roman Empire. And the statistics are just, they're staggering.
They really are.
This isn't just a poisonous mushroom. It is the poisonous mushroom.
He was the king of toxicity.
Mm.
I mean, if you look at the global statistics for mushroom-related fatalities, Amanita Phalloides is responsible for approximately 90% of them.
90.
90.
90%. It's an overwhelming majority. It's a biological superpower. You have thousands of mushroom species, many of them toxic, but this one single species is doing almost all of the killing.
And what really struck me reading through the descriptions in the source material is this idea of visual deception. It's profound. I think when we imagine a poisonous mushroom, we all have this cartoon image in our heads.
Oh, yeah. The Super Mario mushroom.
The red one with the white spots, right? The Amanita muscaria, or the fly agaric. Exactly. And that mushroom, it screams, warning. It's bright red. It's flashy. It can be hallucinogenic. It signals danger from a mile away.
But the death cap.
The death cap does the exact opposite. It's unassuming. It has this cap that can range from a sort of greenish olive to a bronze, or sometimes it's completely white.
So it's variable.
Very variable. And have a white stem, white gills. It looks edible. It looks like something you would actually want to put in a pasta sauce.
It looks harmless. And that is the paradox we're really exploring today. How does something so unassuming manage to be such a perfect, and I mean perfect, killing machine?
Right.
And we aren't just going to look at the toxicology, though we have plenty of that. We're going on a bit of a journey today because the mission of this deep dive is to really understand the death cap's success.
It's a success story, in a way. A terrifying one.
A terrifying one, yeah. We need to look at how it invades new continents, how it chemically tricks the human body, and the incredible genetic detective work that revealed how it's, well, conquering the west coast of North America.
It's an invasion story as much as it is a toxicology story. And it's a story of deception on multiple levels: visual deception, biochemical deception, and finally, clinical deception. The mushroom lies to your eyes, then it lies to your cells, and finally, it lies to your doctors.
So let's start where the mushroom starts, in the woods. If I'm walking through a forest, say in Northern California or somewhere in Europe, and I look down, what am I actually seeing? Because the sources are clear that identifying this thing isn't as straightforward as just looking at the color.
Right. And this is where the danger really lies for foragers, amateur, or even experienced. The morphology, the physical structure of Amanita Phalloides is distinct, but it shares features with perfectly edible species. The cap is usually pretty substantial, about 5 to 15 centimeters across. And like I said, the color is all over the place: pale green, olive brown, white. But one of our texts noted a specific tactile feature. The cap is sticky when it's wet, and the skin peels really easily.
Which is terrifying because I've heard amateur foragers use that as a rule of thumb. You hear these rhymes, if it peels, it's a meal.
That is a dangerous, potentially fatal myth.
Yeah.
The death cap peels beautifully. It feels satisfying to handle.
Yeah.
Then below the cap, you have the gills. In the death cap, they're white and they're free, which is a mycological term meaning they don't attach to the stem.
Okay, so white, free gills?
But the most critical feature, the absolute smoking gun for identification, is at the very, very bottom of the mushroom. It's called the vulva.
The cup of death, as one of the sources called it.
A bit dramatic, but
Yeah.
it's accurate.
Yeah.
The mushroom grows out of something called a universal veil, which you can think of as a protective egg. As the mushroom pushes up and grows, it breaks that veil. And the remains of that veil form a ragged, sac-like cup at the base of the stem. But here's the problem. That cup is often buried underground or hidden under leaf litter.
Ah, so if you're out foraging and you just walk up and cut the mushroom at the soil line with a knife,
You miss the warning sign entirely.
you leave the evidence
You
behind.
leave the evidence in the dirt. What you're holding in your hand is a mushroom with a ring on the stem, white gills, and a nice, unassuming cap. To the untrained eye, it looks generic. And in this case, generic gets you killed.
And then there's the smell. You'd think something called the death cap would smell like, I don't know, rotting meat or sulfur.
Initially, no. And this is another part of the deception. The sources describe the scent of a young, fresh death cap as faint and honey-sweet,
Honey sweet.
pleasant. It invites you in. It's only as the mushroom ages and begins to decay that the smell becomes overpowering and kind of sickly sweet or foul. But by then, it's usually too old for anyone to want to pick it for food anyway. It traps you when it's fresh.
That honey sweet detail gave me chills. It's like every part of it is designed to be alluring. And speaking of traps, we have to talk about the taste. The delicious trap, as the outline calls it. There's this pervasive idea that poisons taste bad, that nature has a built-in bitter setting for things that will kill us.
That is another fatal misconception.
Mm-hmm.
We have reports from survivors and unfortunately from the history books regarding those who didn't survive. And they consistently report that the fungi tasted excellent.
Not just okay, but excellent.
Delicious. There is no burning sensation, no bitterness, no immediate gag reflex. You eat it and you enjoy it. The poison gives no warning on the palate.
And that really, really complicates things for immigrant populations, doesn't it? I was reading about the demographics of poisoning cases, particularly on the west coast of the U.S. and in Australia. And it seems like there's this tragic pattern of cultural confusion.
It is a major, major issue. You have people coming from Southeast Asia, from Laos, Cambodia, China, who are expert foragers in their home countries. I mean, these are people who have been picking wild mushrooms their whole lives.
So they know what they're doing, or they think they do.
Exactly. In Southeast Asia, there is a very highly prized edible mushroom called the paddy straw mushroom, Vulvariella vulvacea. It's a staple.
And I'm guessing it looks like the death cap?
In its early stages, yes.
Yeah.
When the paddy straw mushroom is in its little egg stage before the cap fully opens, it looks remarkably similar to the young egg stage of Amanita phalloides.
Oh, no.
And tragically, the death cap also resembles another edible favorite, the white Caesar mushroom, Amanita princeps. So you have people applying their indigenous knowledge, their perfectly valid expertise, to a new landscape, and they don't realize that the rules have changed, that a lethal look-alike is waiting for them here.
So it's not ignorance, it's a mismatch of ecological context.
Precisely. And we see the exact same thing in Australia. Immigrants mistaking Amanita phalloides for the edible species they know and love back home. It's just heartbreaking because these are often communal meals. A family dinner turns into a mass casualty event.
So let's go there. Let's say the worst happens. Someone has mistaken this for a paddy straw mushroom. They've taken it home, sautéed it, maybe with some garlic and butter, and they've eaten it. What is happening on a molecular level? Because the source material from stat pearls goes into this biochemical warfare in incredible detail, and it is fascinatingly terrifying.
It really is.
First off, can't you just cook the poison out, boil it?
Absolutely not. That is the first and maybe most important weapon in its arsenal. The primary toxins are called amatoxins, and these amatoxins are thermostable.
Thermostable, meaning heat doesn't affect them.
Correct. You can boil them, you can sauté them, you can freeze them, you can dry the mushroom out and store it for 10 years. The structure of the toxin is a ring-shaped peptide, that sort of loop of amino acids, and it is incredibly tough. It does not break down with heat.
So you ingest it, it goes into your stomach.
Right. And actually, there are two main groups of toxins in the mushroom. There are phallotoxins and amatoxins. The phallotoxins are nasty, they're toxic to liver cells, but interestingly, our gut doesn't absorb them very well.
Okay, so they're not the main problem.
They might contribute to some of the initial gastrointestinal distress, but they aren't the killers. The killers are the amatoxins, and specifically a molecule called alpha-amanitin.
Alpha-amanitin, that's the assassin.
That is the assassin. It is absorbed rapidly by the intestine, and then it's transported through the bloodstream directly to the liver.
Of course, the liver, the body's detox center.
Exactly. It's the first line of defense, so grabs these toxins out of the blood first. It uses specific cellular doorways called OATP transporters. So the liver essentially opens the door and invites the assassin right inside.
And once it's inside the liver cell, what does it do? What's its target?
It targets a very specific and absolutely vital enzyme called RNA polymerase II.
Okay, RNA polymerase II. That sounds complex.
It is, but we can simplify it. Think of RNA polymerase II as the factory foreman in every single one of your cells.
Okay, the former.
Its job is to read your DNA and transcribe it into messenger RNA, or mRNA. And mRNA is the blueprint that the cell uses to build proteins.
So if you stop the foreman.
The entire factory stops.
Mm.
No mRNA means no new proteins. And your cells are constantly repairing themselves, building new enzymes, maintaining their structure. They need to make proteins to live.
So you're saying this toxin just, it grinds the entire machinery of life to a halt at the source code level?
That's a perfect way to put it. Cell metabolism just stops. The cell essentially starts to self-destruct. It's a process called necrosis. The liver cells just start dying off en masse.
Wow. And here's a detail from the research that blew my mind. The mushroom doesn't poison itself.
Right. Isn't that incredible? The mushroom itself has RNA polymerase, obviously, because it needs to grow and make its own proteins.
Yeah.
But the mushroom's own version of that enzyme is immune to the toxin. It has a slight structural difference that prevents the toxin from binding to it. So the mushroom carries this absolutely lethal weapon, but has a built-in shield. It's walking around with a grenade that it can't detonate on itself.
That is just, it's diabolical evolutionary engineering.
It really is.
Okay, so the cells are dying, the liver is under attack. But the scariest part of this, reading the clinical timeline, is that the person who ate the mushroom doesn't know any of this is happening. Not yet.
This is the clinical horror story, as you put it. The poisoning progresses in 4 distinct and very cruel phases, and the timing is what makes it so deadly. Phase 1 is the silent period, the incubation.
Silence.
Complete silence. For 6 to 12 hours, sometimes up to 24 hours, the victim feels perfectly fine. They've had their meal, they feel great, they might go to sleep, wake up the next day, go to work.
But the liver is already under a full-scale assault.
Yes. The toxin is in the blood, it's being absorbed by the liver, it's binding to the enzymes, but the cellular death hasn't reached a critical mass that causes symptoms yet.
And that delay is catastrophic for treatment.
It's the whole ball game. If you feel sick immediately after eating something, you go to the ER, they pump your stomach, they give you charcoal, you have a chance. But if you wait 12 hours, the toxin is already fully absorbed. The window for that early decontamination is slammed shut.
And then after that silence comes phase 2.
The gastrointestinal phase. And it hits like a freight train. Sudden, severe, carky abdominal pain, nausea, vomiting, and a profuse watery diarrhea that the medical texts often describe as cholera-like.
So it's violent.
Extremely violent and exhausting. The major risk in this phase is severe dehydration and electrolyte imbalance, which can be dangerous in itself.
But, and this is the part of the story that just feels like a psychological thriller, it stops.
Phase 3, the latent period. We often call it the walking ghost phase.
The walking ghost.
After about 24 hours of those horrible GI symptoms, they just subside. The vomiting and diarrhea stop. The patient feels better. They might sit up in bed, ask for food, maybe even get discharged from the hospital if the doctors aren't aware that mushrooms were involved.
That is a chilling term. Because they aren't actually better, are they?
No. It is the mushroom's cruelest trick. While the gut has settled down, the liver is quietly being destroyed. If you were to look at their blood work during this recovery phase, their transaminase levels, those are the enzymes that leak out when liver cells burst, are skyrocketing.
On paper, they're dying.
On paper, their liver is dissolving, but the patient feels a sense of relief. It's a false dawn.
And then the crash, phase 4.
Terminal organ failure.
Yeah.
This usually happens 3 to 7 days after the mushroom was eaten. The liver fails completely. This leads to jaundice, the yellowing of the skin and eyes. You get coagulopathy.
Meaning the blood stops clotting?
Right. The liver isn't making clotting factors anymore, so you have spontaneous internal and external bleeding.
Yeah.
And then hepatic encephalopathy.
Which is when the toxins that the liver should be clearing out hit the brain.
Exactly. The liver normally filters ammonia and other toxins from the blood. When it fails, those toxins accumulate and reach the brain. It causes confusion, delirium, agitation, and eventually coma.
And the kidneys often fail too, right?
Yes, that's hepatorenal syndrome. The kidneys can fail either from direct toxicity or just secondary to the complete collapse of the liver. Without aggressive intervention, the mortality rate historically was 60 to 70%. Even today, with modern medicine, but without the right specific treatment, it still ranges from 10 to 30%.
It's a total system collapse, a cascade of failure. And it all started with a tasty meal 3 or 4 days prior.
Incredible, isn't it?
It is. So let's talk about hope. Let's talk about the race for a cure, because the sources mention some very specific and some surprisingly ancient interventions. It's not a complete death sentence if you get to the hospital.
No, it's not. The first line of defense is just good old-fashioned supportive care. You have to keep the patient hydrated to counteract that violent GI phase. But there is a specific physiological loop we have to break called enterohepatic circulation.
Okay, unpack that for us. Enterohepatic. So gut and liver.
Precisely. So the body is smart, or tries to be. The liver grabs the amatoxin and tries to get rid of it by dumping it into the bile. The bile gets secreted into the intestines to help with digestion.
Okay, so it's trying to excrete it.
But here's the loop. The gut, detecting these chemicals, reabsorbs them and sends them right back to the liver through the portal vein.
It's recycling the poison.
It creates a cycle of repoisoning. The toxin goes round and round from liver to gut, back to the liver, doing more damage each time.
So how do you stop it? How do you break that loop?
Activated charcoal. But not just one dose. You give it repeatedly. The charcoal sits in the gut and acts like a sponge. So when the bile dumps the toxin in, the charcoal binds it and prevents it from being reabsorbed. It effectively breaks the cycle.
But that's just containment, right? That's damage control. What about an actual antidote? The sources mention penicillin, which really surprised me. I think of it for bacterial infections.
It is surprising. High-dose intravenous penicillin G has been used for a long time in these cases. It's not killing bacteria here, it's actually competing with the toxin. It's thought to try and occupy those OATP transporters, the doorways into the liver cells, to block the toxin from getting back in. But it's not the most effective option we have anymore.
No, the real star of the show seems to be something derived from a plant, the blessed milk thistle.
Yes, Silybum marianum. The compound is called silibinin, and there is a specific intravenous pharmaceutical formulation mentioned in the Mylan clinical trial protocols called Legalon SIL. This is the gold standard for treatment right now.
So how does the thistle fight the mushroom? What's the mechanism?
It works on two fronts, which is what makes it so effective. First, like penicillin, it blocks those OATP transporters. It essentially locks the doors to the liver cells so any amatoxins still floating in the blood can't get back in to do more damage. Okay, that's defense.
But secondly, and this is key, it stimulates RNA polymerase first.
Not polymerase II, which the toxin blocks.
Exactly. It stimulates a different polymerase that helps the liver tissue regenerate. It encourages the synthesis of new cellular machinery in the surviving cells. So it's playing defense and offense. It's protecting the surviving cells while also helping them rebuild.
And the results are good.
The data from the uncontrolled trials we have is incredibly promising. The reports suggest mortality drops to less than 10% with silibinin compared to over 20% with penicillin G.
That is a massive difference. That's saving a lot of lives. And the Mylan protocol mentioned in the sources is essentially trying to formalize this into a global standard. Right?
Exactly. The protocol involves a big loading dose followed by a continuous intravenous infusion for days. It's an aggressive treatment because you are literally in a race against necrosis.
And if silibinin fails, or if the patient just comes in too late, say, they're already deep into phase 4, then the only option left is a liver transplant. You simply have to replace the destroyed organ. There's no coming back from that level of damage.
Which is a high-stakes, lifelong consequence. You're trading a mushroom poisoning for a lifetime of immunosuppressant drugs.
Correct. It saves your life, but your life is changed forever.
Now, I want to pivot, because this is the part of the story that I found just as compelling as the toxicology. We've talked about how it kills, but why is it here? Specifically, why is it here in North America? Because, as the sources point out, Amanita Phalloides is a European fungus.
It is. It belongs in Scandinavia, in the UK, in the Mediterranean. It's European native.
So how did it get to California, and what is it doing there? This brings us to the really cool genetic detective work from the Fungal lab.
This is a fascinating piece of biological forensics. So first, you have to understand that Amanita Phalloides is an ectomycorrhizal fungus.
Okay, that's a big word. Ecto. Mycorrhizal.
It just means it needs a tree. It forms a symbiotic relationship. It wraps its mycelium, its root-like network, around the roots of a tree. The fungus is great at pulling nutrients like phosphorus from the soil and giving them to the tree. In return, the tree gives the fungus sugars from photosynthesis.
They're partners.
They're partners. They cannot survive without each other, which is a key clue to how it got here.
Right. It can't just float across the Atlantic Ocean on a piece of driftwood.
No, and its spores don't travel thousands of miles on the wind. It needs a living host to travel with. So It arrived with humans.
Hitching a ride.
Exactly. Hitching a ride on the roots of imported exotic trees, cork oaks, chestnuts, European pines, that people brought over in the 19th and 20th centuries for landscaping.
And the Pringle lab actually pinpointed the patient zero locations in California, didn't they? By looking at old samples?
They did. They went through historical collections in University Herbaria. And the first confirmed California collections were in 1938 at the Del Monte Hotel in Monterey, and then again in 1945 on the UC Berkeley campus.
And both of those places are famous for importing exotic trees for their grounds.
Famous for it. The Del Monte Hotel had these grand European-style gardens. UC Berkeley is a world-renowned botanical garden. It fits the story perfectly. The fungus came over in the soil of these potted saplings from Europe.
But here's where the genetics get really, really interesting, because usually when a species invades a new place, you have what's called a founder effect or a bottleneck.
Right. A few individuals arrive, so the genetic diversity is really low. It's like a shallow gene pool.
And the Pringle lab found that, yes, the California populations are genetically less diverse than the European ones overall, which you'd expect, but there's a huge twist. They found that the California population is a melting pot of death caps. What does that even mean?
Well, in Europe, the populations are somewhat genetically distinct by region. You have, for example, a Norwegian type of death cap. You have a French type, a Corsican type. They tend to stay in their own region.
They stick to their own turf, so to speak.
Exactly. But in California, because we imported trees from all over Europe and planted them all together, we brought all these different genetic lineages to the same place.
So you're saying in a park in Berkeley, you might have a death cap with Norwegian ancestry growing right next to a death cap with Corsican ancestry.
Precisely. You can find them in the same spot, under the same tree, and crucially, they are mating. They're mixing their genes in ways that just don't happen in their native range.
And that creates new combinations.
It's creating novel genotypes.
Yeah.
And the implication, the theory, is that this melting pot could be creating a super adapted version of the fungus.
Yeah.
One that's more vigorous or more adaptable to the California climate than its ancestors were. We may have accidentally created a stronger invader.
That leads perfectly into the next section on its ecological behavior, because something really alarming is happening on the West Coast. This fungus didn't just stay in the manicured gardens of the Del Monte Hotel. It jumped the fence.
This is the host shift. And from an invasion biology perspective, it is the nightmare scenario.
Yeah.
Because usually, these mycorrhizal fungi are loyal. They're specialists. A pine mushroom stays with pines. A birch bolete stays with birch trees. But the death cap in California did something different. It learned to bond with a native tree, the coast live oak, Quercus agrifolia.
So it moved from the imported cork oak to the native California oak.
Yes. And once it made that jump, it was free. It didn't need the curated gardens and parks anymore. It could move out into the wild, undisturbed forests of the West Coast, like Point Reyes National Seashore.
And it is spreading. The numbers in the report were surprising. I saw an estimate of 5 to 9 kilometers per year.
That is incredibly fast for a mycorrhizal fungus. For comparison, many invasive plants only move about 2 kilometers a year. This fungus is racing through the soil from oak tree to oak tree. It is the only known invasive ectomycorrhizal fungus in North America.
And it's not just existing there in the wild. The research says it's dominating.
Right. In some study sites in California, the death cap is now the most abundant mushroom in the forest, measured by biomass. It is outcompeting the native fungi.
So it's displacing them.
It's taking up the root space. It's forming more connections. We don't fully know what the long-term ecological impact will be, but it is effectively rewriting the fungal map of the West Coast.
It's a silent conquest happening under our feet.
Right.
This European invader is just taking over.
And because it has successfully associated with native trees, there's really no way to stop it. We can't cut down all the coast live oaks in California. The death cap is here to stay. It's a permanent resident now.
It really puts a new perspective on the term invasive species. We think of weeds we can see or maybe feral pigs, but this is a hidden invasion.
It connects right back to that visual deception we started with. It looks like it belongs there now. It grows under the native oaks. Unless you were a trained mycologist, you wouldn't know you're looking at an alien invader. You'd just think, oh, a mushroom in the woods.
We can't do a deep dive on the death cap without touching on its role in human history, because this mushroom has shaped major events in a very literal way. The sources mention the Holy Roman Emperor Charles VI.
1740, a pivotal moment in European history. Charles VI, the emperor, ate a dish of sautéed mushrooms. He fell ill. The reports say indigestion first, then a delay, then he died about 10 days later.
That timeline, it matches the amatoxin profile perfectly.
It does. Phase 1, the silent period. Phase 2, the GI distress. Phase 3, the false recovery. And Phase 4, organ failure. And the consequences of his death were massive.
They had no male heir.
No male heir. His death ended the direct male line of the House of Habsburg. This sparked the War of the Austrian Succession, which reshaped the map of Europe. The philosopher Voltaire famously wrote, "This mushroom dish has changed the destiny of Europe."
That is mind-blowing. A single fungal fruiting body literally changed the borders of nations.
And then if you go back even further, there is the Roman Emperor Claudius in 54 AD. He was known to love Caesar's mushrooms, Amanita Phalloides, which are a delicious, safe, edible amanita.
Right.
The historians Tacitus and Suetonius suggest that his wife Agrippina, who wanted her son Nero to take the throne, had the chef slip some death caps into his meal.
Or maybe used a concentrated juice or poison made from them.
That's another theory. Either way, he died, Nero became emperor, and we all know how that went. Rome burned. Again, the death cap potentially pivoting the entire course of Western history.
But it's not just ancient history. That's what's so chilling. The sources bring up a very modern, very recent case, the Lichen mushroom murders in Australia in 2023.
This was a huge international news story. A woman named Erin Patterson served a beef Wellington to her former in-laws. It allegedly contained death caps.
And the result?
3 of the guests died. A 4th survived only after receiving an emergency liver transplant. It highlights that this isn't just a folklore danger or a Roman intrigue. It is a modern forensic issue. It's a weapon that is easy to find, difficult to trace, and devastatingly effective.
And so difficult to diagnose until it's far too late.
Exactly.
So we've covered a lot of ground here. We've gone from the deceptive white gills to the shutdown of RNA polymerase, from the walking ghost phase in the hospital to the genetic melting pot of California. If we had to distill this all down for our listener, what are the big, crucial takeaways from this deep dive?
I think, for me, the first and most important is the theme of deception. You have to understand that this mushroom does not look, smell, or taste like a killer. And that ghost phase in the hospital is the most dangerous part of the poisoning because it gives everyone false hope. You cannot trust how you feel. You have to trust the toxicology.
And the genetics piece, that was a revelation for me.
The melting pot. We are witnessing evolution in real time. By bringing trees from all over Europe and planting them side by side in California, we inadvertently created a laboratory for this fungus to mix its lineages and potentially become a stronger, more resilient invader. It's a powerful lesson in the unintended consequences of globalization.
And finally, that leads to the invasion.
The fact that it host shifted. The moment it learned to love American trees, the game changed. That means it's not just a visitor in our gardens anymore. It's a permanent resident of our wild forests. It's traveling up to 9 kilometers a year through the soil, and there is no putting that genie back in the bottle.
It really changes how I look at the forest floor. You know, we often think of nature as this fragile thing that needs our constant protection. But here's an organism that is robust, incredibly adaptable, and, well, ruthlessly efficient.
It commands respect. That's the word I always come back to. We don't need to fear the woods, but we absolutely need to respect the powerful chemistry that exists there.
So here's a final thought to chew on. We tend to view the death cap as a villain. It's right there in the name death cap. But from the mushroom's perspective, it doesn't want to kill us.
No. We are a complete dead end for it.
Right. If a human eats a mushroom, the spores don't get spread. They get destroyed in our stomach or, trapped in a coffin. We are not part of its life cycle at all.
We are irrelevant.
We're totally irrelevant. It probably developed those toxins to deter insects or slugs, or maybe for no reason at all. Maybe it's just some random metabolic drift. We are just profoundly unlucky enough to have a liver with an enzyme that happens to be the perfect target for its particular brand of poison.
We are just collateral damage in its quiet, relentless quest to colonize the roots of the world. It's a humbling thought, isn't it?
It really is. So, to everyone listening, enjoy the woods, admire the mushrooms, photograph them. They are incredibly beautiful. But unless you are 100% sure, and I mean expert level, bet your life sure, keep them off your plate. And remember, the sources are clear that even tasting a tiny piece of a death cap and spitting it out is a gamble you absolutely should not take.
Absolutely. Leave the identification to the experts and the eating to the grocery store unless you truly know what you are doing.
Stay curious, stay safe, and we will catch you on the next deep dive.
Take care.